In GCSE Biology, inheritance, variation and evolution is a really tricky topic that often trips students up. This topic is hard because it introduces loads of new vocabulary, specific scientific terms like natural selection, alleles and mitosis that can sound a bit scary and confusing. It also includes some quite complex processes like evolution and genetic engineering, and even requires some maths like calculating probabilities. But don't worry, I'm here to help. In this video, I'll walk you through the whole topic so you can leave with a really clear picture of how it all fits together and exactly what you need to know for the exam. We've already got revision notes on the Save My Exams website for the details. This video overview is about seeing how inheritance, variation and evolution all fits together. This topic starts with one question. How does genetic information get passed from parents to offspring? And the answer comes down to a specific type of cell division, meiosis. You absolutely must know the difference between mitosis and meiosis because exam questions love testing this. Mitosis produces two genetically identical cells. It's for growth, repair and asexual reproduction. Think cloning, exact copies. Meiosis produces four genetically different cells, each with half the number of chromosomes. It happens in reproductive organs to make gametes, sperm and egg cells. This is important because when gametes fuse at fertilization, you get the full chromosome number back. It's because of meiosis that every offspring is genetically different. That variation, that's what makes evolution possible. So remember, mitosis equals identical, meiosis equals variation. Now, let's zoom in to the molecular level. DNA is a double helix made of nucleotides. Each nucleotide has a sugar, a phosphate, and one of four bases, A, T, C, or G. The bases pair up in a specific way, A with T, C with G. That's complementary base pairing, and you must know it for the exams. Higher tier students, pay attention. You need to know how DNA codes for proteins. Every three bases code for one amino acid. The order of bases determines the order of amino acids, which determines the protein that gets made. The process works like this. A template, mRNA, carries the code from DNA in the nucleus to ribosomes in the cytoplasm. Carrier molecules bring amino acids to the ribosome in the right order. The chain folds into a specific shape, and that shape is very important. An enzyme's shape determines whether it works. A structural protein shape determines its strength. Change one base and you might change the whole protein, which brings us to mutations. Mutations are random changes in DNA. Most do nothing. A few change the protein slightly. Very rarely, a mutation creates something beneficial. Now, here's where students get confused. Genotype versus phenotype. Your genotype is the alleles you have. An allele is just a particular version of a gene. So your genotype is the actual DNA code. Your phenotype is what you can see, your characteristics. For example, you might have the genotype big T, little t for height in pea plants. The phenotype, tall, because the allele we are calling big T, tall, is dominant over the allele we are calling little t, short. You need to be comfortable with these terms. Homozygous equals same alleles, genotype big T or little t, little t. Heterozygous equals different alleles, genotype big T, little t. Dominant equals the allele is always expressed, even with just one copy, and recessive means the allele is only expressed when two copies of it are present. I'm sure you've noticed that reproduction and inheritance contain so many new keywords that you need to learn. On the Save My Exams website, you can find loads of flashcards on these topics to help you learn the key terminology with confidence. Punnett Squares let you predict offspring outcomes. Here's an exam tip. Always write the dominant allele first, so big T, little t, not little t, big T. Use letters where the capital and lowercase are obviously different, like big A and little a, not big C and little c. You need to be able to calculate ratios, like 3 to 1 tall to short, and probabilities, like 25% chance of being short. Higher tier students might need to construct these crosses from scratch, so make sure you practice that. Two inherited genetic disorders come up constantly: polydactyly and cystic fibrosis. Polydactyly, extra fingers or toes, is caused by a dominant allele. So if one parent has it, there's a 50% chance the child will too. Cystic fibrosis is caused by a recessive allele. Both parents can be carriers, big F, little f, and completely healthy, but there's still a 25% chance their child will have the disorder, little f, little f. This has huge implications for embryo screening and gene therapy. These topics raise ethical questions you might be asked about. So now we know how characteristics get passed on. The next question is, how do populations change over time? Variation means differences between individuals. It can be genetic, like blood group or eye colour, or environmental, like scars or language. Or it could be a combination of both, like height. You inherit tall genes, but a poor diet can stunt your growth. All genetic variation comes from mutations. Remember, mutations are continuous and random. Most have no effect, but occasionally a new allele gives a survival advantage. That's where natural selection kicks in. Evolution is a change in inherited characteristics over time. Natural selection is the mechanism that causes evolution. Here's how it works. Variation exists in a population due to mutations. Individuals with characteristics best suited to the environment survive. They reproduce and pass on advantageous alleles. Over many generations, those alleles become more common. Here's an exam tip. Don't confuse evolution, the change itself, with natural selection, the processes that causes the change. A classic example of evolution is antibiotic resistance in bacteria. Bacteria reproduce every 20 minutes, so evolution happens fast. A random mutation makes some bacteria resistant to antibiotics. When we use antibiotics, the resistant ones survive. They reproduce without competition. Soon the whole population is resistant. This evolution is happening in real time, and that's why we must use antibiotics carefully. Always finish the course of antibiotics, don't use them for viral infections, and limit their use in agriculture. Humans have been influencing evolution for thousands of years through selective breeding. The process is simple but slow. Choose parents with desired characteristics, breed them together, select the offspring with those desired characteristics, and then repeat this for many generations. This has given us high-yield crops, disease-resistant plants, and gentle dogs. But there is a problem: inbreeding. When you only breed the best individuals, you actually reduce the gene pool. This increases the risk of genetic defects and makes populations vulnerable to new diseases. Genetic engineering is a more precise process. Instead of waiting for generations, scientists cut out specific genes and insert them into other organisms. Higher tier students, you need to know this process. Restriction enzymes cut out the gene, leaving sticky ends. The same enzyme cuts a bacterial plasmid. DNA ligase then joins these together. The recombinant plasmid goes into bacteria, which then produce the protein, like human insulin, for diabetes treatment. GM crops can be herbicide resistant or pest resistant, improving yields, but concerns remain about effects on wild insects and long-term health impacts. Then there's cloning, producing genetically identical organisms. Methods include tissue culture for plants, embryo cloning for animals, and adult cell cloning. Each method has benefits, like preserving rare species or producing disease-resistant crops, but there are also risks, like reduced genetic diversity and possible ethical concerns. Questions asking you to evaluate the benefits and risk of things like genetic engineering or cloning can be quite difficult. But on the Save My Exams website, we've got loads of exam questions that will help you to practice balancing arguments for these kinds of topics. When it comes to genetics and evolution, the exam won't just test scientific facts. It'll also test your understanding of how those facts were discovered. Charles Darwin proposed natural selection after a round-the-world expedition and years of research. His theory states that individuals show variation, those best suited to the environment will survive and reproduce, advantageous characteristics then get passed on to offspring, and over time the species evolves. But his theory was slightly controversial. For example, it challenged religious beliefs about creation. There also wasn't enough evidence at the time, and crucially, nobody actually knew how inheritance worked. DNA wouldn't be discovered for another 50 years. Another guy called Alfred Russel Wallace independently developed the same theory. The two published together in 1858, and then Darwin published his famous book on the origin of species in 1859. Wallace's key contributions included his work on warning coloration, like bright colours that deter predators, and speciation, how new species form. Speciation happens when populations become isolated, e.g. by a mountain range or river. The populations face different environmental pressures, and eventually they evolve so differently they can no longer interbreed to produce fertile offspring. You need to be able to compare this with Lamarck's theory, which was wrong. Lamarck thought that characteristics acquired during life could be inherited, like giraffes stretching their necks and passing on longer necks to their offspring. We now know this doesn't happen. Changes must occur in DNA in order to be inherited. Before Darwin, Gregor Mendel was breeding pea plants and discovering patterns of inheritance. He showed that hereditary units, we now call them genes, are passed from parents to offspring unchanged. He demonstrated that some units are dominant over others, but his work was ignored during his lifetime because it was completely new. Nobody knew about DNA, genes or chromosomes yet, and the mechanism made no sense to scientists at the time. Only in the early 20th century, when scientists observed chromosome behaviour during cell division, did Mendel's work make sense. Chromosomes behaved exactly like his hereditary units. By the mid-20th century, DNA structure was determined and the gene theory was complete. We now have multiple lines of evidence for evolution. Fossils show how organisms have changed over millions of years. They form when organisms are preserved in rocks, either as actual remains, if decay conditions are absent, as mineral replacements, the kind of fossils we usually see, or as traces, like footprints. The fossil record is incomplete because soft-bodied organisms rarely fossilise, and geological activity destroys many fossils. That's why we can't be certain how life began. Antibiotic resistance is evolution we can observe. Bacteria evolve resistance through random mutations, and when we use antibiotics, we create selection pressures that favour resistant strains. MRSA is a dangerous example. It has evolved resistance to most antibiotics, making infections very difficult to treat. This is where our SmartMark AI tool can be really helpful. An exam question might ask you for evidence of evolution. SmartMark gives you instant feedback on whether your explanations are detailed enough for full marks, and even gives you guidance on how to improve your answer. The final piece of the puzzle is organising all of this diversity. Carl Linnaeus created a classification system based on structure and characteristics. The sequence goes kingdom, phylum, class, order, family, genus, species. Remember it using King Philip came over for Gran's spaghetti, or kittens pounce clumsily on furry green spiders. Organisms are named using the binomial system, genus with a capital letter, then species with a lowercase, in italics, like Homo sapiens. As microscopes improved and DNA analysis became possible, a different, much more recent Carl Woese, proposed a level above kingdoms, domains. The three domains are archaea, primitive bacteria found in extreme environments, bacteria, true bacteria, and eukaryota, everything with a nucleus, including protists, fungi, plants, and animals. This system reflects evolutionary relationships more accurately than Linnaeus' system. Evolutionary trees are diagrams that show how species are related over time. Reading them is a key skill. The closer two species are on the tree, the more recent their common ancestor. For example, chimpanzees and bonobos share a very recent common ancestor, so they're most similar to each other. Humans share a more recent ancestor with gorillas than with orangutans, so we're more closely related to gorillas. Evolutionary trees are built using DNA analysis for living organisms and fossil data for extinct organisms. DNA comparison is particularly powerful, and the more similar the base sequence is, the more closely related the species. So, to summarise, inheritance, variation and evolution isn't just four random topics, they're all connected. Reproduction explains how genetic information passes from parent to offspring through meiosis, DNA and genes. Variation and evolution explains how populations change over time through natural selection and how humans speed this up through selective breeding and genetic engineering. Development of understanding gives you the historical context, Darwin, Wallace, Mendel, and the evidence that supports the theory. Finally, classification organises all that diversity into systems that reflect evolutionary relationships. Exam questions will test whether you can connect these ideas. That's why understanding the overall structure matters so much. This video gave you the overview, now you need the detail. The links below take you to our full resources, revision notes that explain every concept clearly, flashcards for quick recall of key terms, exam questions organised by topic with smart mark AI feedback, and past papers when you're ready to put it all together. Start a free trial of Save My Exams to test our resources now.